What's up everybody? So in this video we're going to be looking at this teeny tiny cute piggy, but also we're going to be talking about C2.2, the HL version or the HL details that you guys have to know for C2.2. As usual guys, go check out teachme.org. The best resources for IB biology are there just for you guys to use. Awesome notes and lots of questions, okay? Now, for this video, we're going to be talking about this is going to be the contents of this video. We're going to talk a little bit more about neuron impulse propagation, because in the standard level video, we did do some of this, but not to the extent that HL needs to know. So we're going to learn the HL details of this part. Then we're going to do something completely new, IPSPs and EPSPs. You'll see what that is. And we're going to talk a little bit of saltatory conduction. This is something that you guys already kind of covered for SL, but not in as great as detail as you do for HL, okay? So that's why we're going to be talking about this. And then we're going to go into endogenous and exogenous chemicals and how these can affect the function of nerves. So we're going to be talking about things like cocaine and things like that, right? Other chemicals that can affect the way your nervous system works. And then lastly here, we're going to talk about the role of receptors, and we'll see what that means as well. And obviously, we're going to do a couple questions, but if you want a lot more, you need to check out teachme.org. Now, without further ado, guys, let's just get started. So what exactly do our neurons do again? What's their main role? So remember, their main role was to send electrical signals, little charges, through the neuron to the adjacent neuron, and then that neuron will take that signal and carry it further to whatever other place needs it. For example, the brain might decide to want to contract this bicep. So it sends this signal through this neuron, which will be transmitted to the next neuron by synaptic transmission, as we learned in SL, and they will be propagated through this neuron until finally it reaches the muscle, which will stimulate it to contract. The same way, the same thing can happen in the opposite way. Maybe a neuron can pick up a signal from your skin if you got stabbed and there's pain, and takes to that signal and it travels it and it carries it all the way to this neuron, transmits it, and this one will carry it to your brain where you can feel that pain. Okay, now you're aware of that pain. So there's a process that happens, okay, how we carry this signal through the neuron and how we transmit it between two different neurons. Now, in standard level, we did cover how a signal is sent from one neuron to the next. That was called synaptic transmission. Now, we're not going to cover that in any more detail in HL, okay? So no more extra details regarding synaptic transmission. What you learned in SL is what you need to know. Now, remember, for SL, we also learned how the signal is generated and transmitted through the axon, right? How this impulse, this signal, this nerve impulse is propagated or sent through the neuron, through the axon till the end, right? So that it can be sent to the next neuron. That part, we're going to go into a little bit more detail, because there's like a little graph that you guys need to be able to read and understand that happens during this process. Okay, so I'm going to explain the process. So it may feel a bit repetitive, but as I explain it, I'm going to reveal how the graph looks like, and hopefully that way it makes a lot of sense. Okay, so it may feel repetitive, but it is very important to understand this for HL. Okay, so let's just get into it. So here we have our neuron, right? We're looking at the axon. This is the outside of the axon, and this is the inside of the axon. Remember this part here? What is this called, guys? This is your cell membrane, right? Your plasma membrane. And this is made up of what? Your phospholipid bilayer. And embedded in your phospholipid bilayer, you have these protein channels, these protein pumps, right? All of these things. Now, remember, when we're sending this signal that we're talking about is an electrical signal. So to help us understand what's going on, we're going to put this little device here. This little guy is going to be able to measure the membrane potential. What's the membrane potential again, guys? That is the charge difference between the inside and the outside, okay? This little guy will give us some values, some numbers, okay, as we describe what happens here. Now remember, when your neuron is not doing anything, okay? It's resting, okay? It's not receiving any signals. It's not sending any signals. At that state, the membrane potential, we call it resting potential, okay? So the resting potential is the membrane potential when the neuron is not receiving any signals and not sending any signals. So hopefully that makes sense. So what is responsible for determining or making our resting potential? It's this guy. So normally, this guy, our sodium-potassium pump, this is our sodium-potassium pump. Okay, it is going to pump sodiums out and potassiums in. That's why we can see normally when our neuron is not doing anything, outside of the cell, there's a lot of sodium, but not on the inside. On the inside, we have a lot of potassium, but not on the outside, okay? And that's thanks to the sodium-potassium pump that pumps our sodium out and brings our potassium in. Now, you can imagine this guy, remember, he's pumping 3 sodiums out and 2 potassiums in. So he's pumping 3 positive charges out and only 2 positive charges in. That means the outside is going to be much more positive compared to the inside, right? So we can say that the inside of this neuron is negative compared to the outside. So I can put a little charge here on our little device here. The inside is more negative compared to the outside because we're pumping out so many more positive charges than we're pumping in. So that's why you can see I put all these little negative charges. All along at the resting potential, we have these negative charges thanks to our sodium-potassium pump. So let me show you now this little curve, guys. You guys need to know this. So you guys have to recognize a curve. I'm going to build this curve as we explain the process. Right now, I'm just showing you a small part of it. So you can see here, the x-axis is time, milliseconds. So it's super, super fast. When we send these signals, it takes milliseconds, really, really, really short. Not even a second, right? Really, really fast. And this part here, our y-axis, is going to be our membrane potential. So the charge, okay? You can see here, -70, 0, 30, right? Whatever, right? We can see here. So let's start here. When our neuron is in the resting potential, right? When it's not doing anything, what is the membrane potential? The membrane potential is negative. We know that. But what exactly is it? It's around -70. So I put here negative, but you should know that at resting potential, the exact number is around -70. Okay, -70. Now look here. Look here. Why is there a sudden rise? Okay, as time goes on, why is there a sudden bump here? Remember, to be able to send a signal, you first need to receive a signal. So let's say this neuron here is waiting for a signal, and the brain finally decides, okay, I want to contract my muscle, so it sends this positive signal, this positive charge, this nerve impulse, right? So let's say a little positive charge arrives right here, okay, right here at this part of the axon. A positive charge arrives. When this little positive charge is responsible for this little bump. Okay, so this positive charge arriving makes this area a little bit less negative, because remember, the resting potential is -70. So when this positive charge arrives, it pushes it up a little bit. So now it's more like -50. Okay, it's more like -50 thanks to this positive charge. Now, very, very interesting, when it gets to around -50, this sodium channel opens. Isn't that crazy? So this guy, what do we call that when it opens at a certain charge? We call that voltage-gated sodium channel. So this is a voltage-gated sodium channel, meaning it opens in response to a certain charge, a certain voltage. So when this positive charge comes, it brings it to around -50. -50. And that -50 is going to open this voltage-gated sodium channel. Now, when this voltage-gated sodium channel opens, what does that mean? Look, there's so many sodiums on the outside and so little on the inside. So from membrane transport, what do you think is going to happen? These guys are going to diffuse down the concentration gradient, right? From where there's a lot on the outside to where there's a little on the inside. So it diffuses down here nicely. Now, as they diffuse in, we call that process of sodiums coming in depolarization. Okay, so right now I'm not going to put any words here. I don't want to make it too full, but I have a really nice summary page coming soon where you guys can see that. So just pay attention to the process now. I have all the nice words and exact steps written out for you guys soon that I'll show, okay? So these sodiums come in, we call that depolarization. Okay, so if all these positive charges are coming in, okay, what's that going to do to the inside? It's going to make it hella positive. Look here at the graph. So this small bump here was the signal arriving from a previous part, from a previous neuron or from a previous part of the axon, any previous part, okay? This positive charge comes and it opens this voltage-gated sodium channel, which is now open, and now that causes a sudden massive rush in, the diffusion down of these sodiums into the axon, causing a huge spike. So now the inside is actually positively charged. So let me make this. Let me fix this. So when depolarization happens, meaning these sodiums come rushing in, now the inside is very positive. Look, I'm going to change all of these negative signs. It's becoming very positive. Okay, you can see it on the graph. a nice huge spike. We're at like plus 30 now. So the membrane potential now, so the charge difference across the membrane is now plus 30. Now these sodiums, okay, they are not going to be crammed up in here. They're going to keep diffusing down. As more and more come in, these guys are going to be all crammed, so they're going to start spreading. Okay, they're going to start spreading. Look here. They're spreading, diffusing, right? I don't want to put that many sodiums here, but you can see the picture. As they keep coming in by diffusion, these guys are going to spread down. Now, as they spread down, what happens? Okay, all of these areas that were negatively charged previously will now become positively charged. Okay, so they diffuse down. Okay, cool. Now, you can imagine this. As they diffuse, and eventually they'll stop diffusing. They'll get to a certain point, and now it's going to spread out. But luckily, on these areas, we have more of these channels, right? We don't have channels in only one portion. So you can imagine in this area, we'll have maybe another sodium channel, another potassium channel, and maybe also a sodium potassium pump, right? We'll have more of these things all throughout the membrane. And also in this diagram, I'm just putting it on this side. In reality, it'd be an axon is like a tube, okay? So you can imagine these guys are all around the circle of the tube, right? But for the sake of this diagram, I'm just putting them up here. Otherwise, it'd get really crazy looking. Now, so these sodiums, they spread all the way across here, right? Now, remember, these sodiums that are spreading, what does that really mean when they're spreading? That means this positive charge is spreading, because this positive charge was only here. Now look, we've spread this positive charge all the way to here. So this positive charge is due to the sodiums and the potassiums inside. Due to all these positive charges, that is creating this positive charge. Now remember, this guy, what is he? He's voltage gated, and he opens in response to this charge. Remember, when these sodiums diffuse all the way down here, now this area is going to undergo an action potential. We just showed how this area underwent an action potential. Now this little positive charge is going to stimulate this one to open, right? Because it's voltage gated. So now this positive charge arrived, now this one opens. And you can imagine, right, outside here, of course, there's going to be a lot of sodium, okay? Because all the sodium from here went in, but there's still a lot of sodium over here that didn't go in, right? On the next segment of the neuron. So all of these sodiums are going to now this thing is open, right? Because the sodium discharge came. Now the sodiums are going to come rushing in again, okay? All the way rushing in, causing a peak, right? On the next segment of the neuron, the next segment of the axon. So they come rushing in. Now these areas are going to now be turned positive. And now again, as they spread out all across, right? I'm not going to go too much detail here, but they will spread across, and then there will be more channels here, which will be activated. The voltage gated sodium channels will be activated, and more sodium will come in. And you can see, just like that, we spread the positive charge all the way from here down the entire axon. You can imagine all the way down to the end, right? But we're not done yet. Okay, so let's say we did now that. We're not done yet. After the sodium comes in, right, we call that depolarization. Now the inside's very positive. Now guess what? This potassium channel, it loves a very high charge. So when it gets to like plus 30, these guys open. So now these guys are going to open, and when they open, what happens? There's a lot of potassium here on the inside and very little on the outside. So guess what? These guys are going to start diffusing outwards, right, through this potassium channel. Now, if all of these positive charges are floating out, what does that mean? That means the inside has now very little positive charge, because all these positive charges are rushing out. So what's going to happen to the graph? It's going to go down again, because now if all of these positive charges are floating out, the inside is becoming negative, right? So now you can imagine this. All of these guys, now the inside, this portion here, after these potassiums go rushing out. By the way, when the potassiums rush out, we call it repolarization. So when sodiums came in, it was called depolarization. When these potassiums rush out, it's called repolarization. And when they rush out, now the inside is going to be negative again. Okay, they go rushing out. Now this inside is negative again, right? And you can see it rushes all the way down, back to around negative 70, right? Maybe even lower, like negative 80, okay? Now what happens? So if this happens, now we have a problem. Now the sodiums are all on the inside, and the potassiums are on the outside. That's the exact opposite that we started with. Remember, at the start, we had all the sodiums on the outside and all the potassiums on the inside. Now we have the opposite scenario. So in order for this neuron, this segment, to be able to receive another nerve impulse, it needs to reset. Okay, so now the signal has been sent to the end, but now it needs to reset so it can receive another signal. Otherwise, we can't contract our muscles again. Okay, otherwise it's game over. So to reset this whole thing, we need the sodium-potassium pump. It's going to take our 3 sodiums, right? Remember by this little beautiful tip, 3 sodiums and then 2 potassiums, and it's going to switch them around. So it's going to bring the potassiums back in and our sodiums back out. Okay? It's going to keep doing that. And now we have our original scenario reset. Look, we still have our, oh yeah, I got to change this. Remember, membrane potential is negative again, right? So you can see, by the way, when this repolarization happens, sometimes it goes very low, like beyond -70. It goes like -90. Okay, and it does that because these guys take so long to close. So when the potassiums rush out by repolarization, they rush out so fast, and this potassium pumps takes very long to close. So too many leave. So when too many leave, it gets to like -90. It gets really low. Okay, but when the sodium potassium pump starts working, it sorts them all out and puts them back to -70. So the normal resting potential. Okay, so you can imagine that's also going to happen here on this next segment. The same process, right? All the sodiums came in, so it rise like this. Now that it's plus 30, these guys are going to open. So this potassiums are going to all go out. If all the potassiums go out, what happens? That's called repolarization. So it's going to go down, and then the sodium potassium pump. Oh yeah. As all the potassiums leave, what happens again to the membrane potential? It goes all negative, right? It goes all negative because now all these positive charges are leaving, and now our sodiums are what? On the wrong side, and the potassiums are on the wrong side. So our pump here is going to go and fix all that, bring the potassiums in and the sodiums out, right? What is that? That's our resting membrane potential. Okay, so now it's ready to receive another signal and reinitiate and do the whole process again. Okay, sending our positive charge all the way down to the end of this neuron. Okay, so that's it. Let's quickly look at this on 2 keywords here. You guys already know what an action potential is. All these events: depolarization, repolarization, the sodium potassium pump reestablishing the resting membrane potential. All of these events is an action potential. Okay, that's very important. Now, we're going to talk shortly about threshold potential in a bit more detail, okay? But basically, the threshold potential is a very key vocab you need to know for HL. This is the minimum potential or the minimum charge you need to initiate an action potential. So remember how before I told you, give me a second. I need to undo some stuff because I want a positive charge. No, never mind. So remember before how I told you a positive charge arrives here that will stimulate the sodium channel to open, so sodiums can rush in. Remember that positive charge, if it is not strong enough, it will not reach the threshold potential. So the threshold potential is around -55. And if that positive charge is not strong enough to bring it to -55, then guess what happens? If that positive charge cannot bring this membrane potential to -55, then these guys can't open. And if these guys can't open, then our action potential can't start. So the threshold potential is a very important vocab word to know, because if this positive charge does not reach the threshold potential of around -55, then we cannot have a signal. So then nothing will happen. It will arrive and then stop right there. No signal will be sent. Okay, that's important to understand. Also, of course, remember that these guys, the sodium potassium pumps, they require ATP because they're pumping things by active transport. Okay. Now, let's quickly look at our summary. I promise you guys, here's the words of all this. We'll rush over this because you guys probably it's drilled into your head at this point, but I think this is a really nice, cool summary that it really explains everything. So here, the -55, that's our threshold potential. So let's look here. What is this number 1? That's our resting state or our resting potential. At this point, all of the channels are closed. Our potassium channel's closed, sodium channel's closed, everything's closed. So when so nothing is happening. The inside is negatively charged because of our sodium potassium pump, right? And it's around what charge is the inside? Around -70 at this point, thanks to our sodium potassium pump. Okay, membrane potential is negative. Then our little positive stimulus arrives to this area, okay? Now, at this point, the sodium channel and the potassium channel is still closed. We're only focusing here on the stimulus arriving. Now, once the stimulus arrives, first of all, the stimulus arriving, right, causes this slight increase in the charge, okay? It makes this axon slightly more positive. Now, if this signal is strong enough, okay, if this signal is strong enough that it reaches -55, now depolarization can happen. Okay, depolarization, right, is when these guys open, the voltage-gated sodium channels open so that sodium can come rushing in. Okay, so if the threshold potential is reached, these voltage-gated channels can open, sodium channels will diffuse into the axon to depolarize the membrane. Very important word. And that will cause the inside to become very, very positively charged, right? We can see it bumps up all the way to here. So remember this threshold potential. It's needed to open these sodium channels, to cause them to rush in and go all the way up to +30. Okay, now when we reach this +30, now this guy is going to close. Our sodium channel is going to close. Remember, it only opens at like -55. So if all these sodiums start rushing in and shoots it up to +30, it's not going to be open anymore. Okay, it's going to close. But at this membrane potential, potassium opens, and when potassium opens and they all rush out, the inside becomes negative again, right? So as these potassiums rush out, that's step 4 here, the membrane potential will go down, 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 even too low. When it goes very, very low like that, we call it hyperpolarization. So repolarization is the process of it coming down, down, down because of potassium leaving. So it's becoming more negative and more negative. But when it goes lower than original, lower than -70, so like here, -80 or -90, that's called hyperpolarization. And that happens because these potassium channels take too long to close. So these potassiums leave, and then this door closes too late. So many potassiums have already left that it's -80. Okay, that's very important. Then lastly, we have our step 5, which is the resetting of our resting membrane potential, right? Because right now we have all these potassiums outside and all the sodiums inside. We got to reset it. So the sodium-potassium pump is going to pump our sodiums out and bring our potassiums back in to reset our membrane potential of around -70. And now the ions are on the right side of the membrane, okay? And now it's reset so we can receive another signal. So resetting is super important to receive another signal, because if we don't do this resetting process, then we're going to be like during this whole time here, right? We can't receive another signal because we're not at the resting membrane potential. So we call this the refractory period. So the whole period where we can't receive another signal because our membrane potential is not at the resting membrane potential, we call that the refractory period. So even if another signal arrives at these stages, it will not be that will not initiate an action potential because we're already busy with one. Okay, so that's important. The refractory period is the area where no new action potential can be made because we're already busy with one. Okay, by the way, the little machine that helps us make this graph is called an oscilloscope. Okay, so an oscilloscope is a little machine that you guys should know about that helps us make this little graph, which shows that shows the relationship between time and the membrane potential as we go through a whole action potential. So that's it. That's the summary. All the words are here. I promise you guys. I hope this is really nice to help summarize it for you guys, and hope it's nice and clear. Let's now go on to the next part. So right now we finished the hardest part of this video, in my opinion, the whole HL detailed aspect with the whole graph and all that of the neuron impulse propagation. So the sending of that signal. Now let's go on to this next part, which I think is pretty straightforward. Shouldn't be too difficult. So what is EPSP and IPSP? So it's basically an what do you call that? Not abbreviation. Yeah, an abbreviation for excitatory postsynaptic potential, EPSP, okay? And basically what this is, if we look at our little graph that we just looked at before, remember how I said there's a little stimulus, a little positive charge coming from the previous neuron or a previous segment of the axon that's trying to cause an action potential on the next segment, right? So there's a little positive charge that arrives. Remember how we said that this positive charge or the stimulus, if it's strong enough to reach the threshold, that can initiate our action potential, right? As we saw. Now, we call that little stimulus, if it brings the charge, the membrane potential, closer to the threshold, we call that an EPSP. Okay, so this here would be an example of an EPSP because it's causing the potential. I didn't spell potential right, did I? Potential. How do you spell potential? I think that's right. Correct me if I'm wrong, but I'm pretty sure that's how you spell potential. So if that stimulus causes the membrane potential to become closer to the threshold, we call that an excitatory postsynaptic potential. So this here is an excitatory postsynaptic potential. It's bringing it closer to the threshold, closer to an action potential. Let me show you a little picture here. So if we have the end of one neuron here, so this would be the terminal end, and this would be the beginning of another neuron, so maybe the dendrites or something like that, right? We remember from SL to send a signal from one neuron to the next, we have that whole sequence of events where these vesicles that contain neurotransmitters will release the neurotransmitters, and these neurotransmitters will go and bind to a receptor. And once it binds to that receptor, remember, that's different from the voltage-gated sodium channels. These are ligand-gated. So once this neurotransmitter binds to it, now it opens. So these guys don't open according to charge. They open according to a little molecule or a ligand. A ligand is a little molecule that binds to it. So when this molecule binds to it, now it opens, and now the sodiums can come in. And if these sodiums come in, what's that gonna do? That's gonna make the inside here a bit more positive. So you can imagine that's gonna cause it to go up and up and up and up. If this positive signal is strong enough, okay, strong enough to reach the threshold, then guess what? Then these guys are gonna open because they are sensitive to charge. So if this signal, this EPSP is strong enough, then these voltage-gated sodium channels will open, and now we'll have what? Depolarization. Well, this whole curve will happen, right? The one we just looked at. So make sure you understand what an EPSP is, okay? So excitatory neurotransmitters, such as these, acetylcholine, what they do is when they bind, is they cause an EPSP. They basically allow positive ions like these sodiums to flow in to cause this EPSP. Now, so you can see this. This is very important. So you can see it's more of an all or nothing kind of situation, as I mentioned before. It's either the threshold is reached and the action potential can happen, or it's not reached and nothing happens, like this. It's close, but it doesn't happen. So you can see it's an all or nothing situation. There's no such thing as a strong signal or strong impulse. It's either yes, there is a action potential, or no, there's no action potential. Okay, there's no such thing as strong or weak. Okay, so when you look at this, is actually kind of tricky to understand, but when someone puts pressure on your hand, so say you hit your hand yourself with a hammer very softly, you will cause those neurons to get activated and send a signal, because they're making, by hitting them, you're causing them to be activated and sending action potentials to your brain, right? So when you hit softly, you're activating only a few neurons, because only a few of them, that hitting of your hand cause only a few of the neurons to have the threshold reached, and so only a few of them were able to generate an action potential to be sent through the neuron to your brain. But if you hit harder, it's not that you're making a stronger action potential. No, you're not making a stronger or bigger action potential. All that's happening is you're activating more neurons. So instead of only activating 5 neurons, causing them to have action potentials, you're activating like 20. And so 20 neurons are now sending a signal to your brain. So it'll appear more painful, not because you made a stronger action potential, just because you made more. So remember, it's an all or nothing thing. It's not that you have a big action potential or a small one. You either have one or you don't. There's no difference in their size. So what makes something more painful or more stronger is the activation of more neurons. Okay, instead of making a stronger action potential. Okay, I hope that makes sense. I understand if it's a little bit tricky. Okay. So right, one potential is brought closer to the threshold. That's an EPSP. So what's an IPSP? The opposite thing. Let me show you. So if we have our little resting membrane potential here, and a stimulus comes, and if this stimulus does the opposite thing, it doesn't bring our membrane potential closer to the threshold. In fact, it brings it away from the threshold. That's going to be called an IPSP, an inhibitory postsynaptic potential. Okay, so obviously in this scenario, you're going to have no action potential for sure. Okay, so let's see how that looks like. So membrane potential is brought further away from the threshold. So if we have, again, same thing, a presynaptic neuron, postsynaptic neuron, in this scenario, we have a little neurotransmitter that's inhibitory. So remember, acetylcholine, when it was released, it caused this postsynaptic neuron to become activated by these sodiums flowing in, creating a positive charge, right? This EPSP, bringing it closer to the threshold, right? But in this scenario, GABA, what it does is it's a kind of neurotransmitter that binds to its little receptor. When it binds to its little receptor, it opens. But the problem is its receptor, its channel that opens, is specific for chlorine ions, not sodiums. Sodiums are positive, so when they go in, the inside's going to become positive and come closer to the threshold. But chlorine, look, it has a little negative charge there. So when this thing opens, chlorine will come rushing in, but chlorine's negative. So if chlorine comes rushing in, then guess what? We're going even more negative. We're going to be like negative 80. Okay, we're going away from the threshold. But what's also interesting is GABA, it has another thing. When these neurotransmitters bind, it causes a bunch of little messages to be sent that opens little potassium ions as well. And remember, potassium ion channels, when they open, potassium rushes out because you have a lot of potassium on the inside compared to the outside. So when these potassium channels open, we have this positive charge leaving. When the positive charge leaves, then the inside becomes more negative. So the same effect will happen. So know that an IPSP can happen in two ways. Either if a negative charge comes rushing in, or when a positive charge leaves, okay, causing this IPSP. So that's pretty interesting, right? So it causes an, what do we call that? When it goes lower than resting potential, we call that hyperpolarization, right? Hyper, even lower. Okay, awesome. Now, one last thing, then we're done with this slide, that's kind of important, is the sum of all stimuli affect the outcome. So when we look at a synapse like this, you're never ever, in reality, you're never ever only sending a positive signal. Like in this scenario, we're showing you how a positive signal is being sent. This acetylcholine binds and sodium comes rushing in. And here we're showing you how a negative signal is sent. The GABA binds and chlorine comes in. In reality, when you look at a junction like this, it's very, very complicated. There's some neurotransmitters will cause this to happen, some other ones will cause this to happen, all in at one junction. So there's a mix of signals. Positive things will come in, negative things will come in, but what determines if an action potential is made is the combination of all of those IPSPs and all of those EPSPs. Okay, there will be some positive signals, some negative signals, but the combination, adding them all together, if they reach the threshold, then you'll have an action potential. Because maybe you have this scenario here, okay, there was some positive signals, but some negative signals, so therefore you just barely didn't make it, okay? So no, it's never just one or the other. It's a combination of many, many stimuli, many IPSPs and EPSPs that affects the full-on outcome, okay? So that's very key to understand. Know these two things. So now we move on to saltatory conduction. This one will be pretty straightforward because we really covered most of it in SL. So remember how in reality, our axons have a lot of these things on them, these little cells called Schwann cells. And when they keep wrapping and wrapping around your axon, you form what is called myelin, which is just a bunch of Schwann cell wrapping around the axon, right? That's our myelin. And what's special about myelin, okay, these little things, these little areas, is that there is no, in these areas, there is no channels, okay? So it's kind of blocking, it's an area where there's no ion movement because there's no sodium channels, no potassium channels, no sodium potassium pumps. So in these areas, there's no ion movement, okay? That's very, very important to understand. So how do these things affect our nerve impulses? Well, they do many things. One of the key things is they speed it up, because you can imagine now when, let's try and make this clear. When we have our sodiums on the outside, right, and they start rushing in by depolarization, right? Let's say they caught rushing in, depolarization, as we just saw, it makes the inside positive. So we have all these positive charges. Now, remember, normally these sodiums would spread out and activate the next sodium channel, right, to cause it to open and then cause that area to have to be open so that sodium can influx and cause depolarization there. Now there's no channels here, so these sodiums are forced to float a little bit further and activate the sodium channels here. Okay, let me make it clear. So basically what these myelin does is it allows us to bypass certain areas. It's areas where there is no channels at all, so nothing can happen here. Therefore, these sodiums have to kind of bypass it and activate the next spot. Okay, now here, if it activates this spot, now sodium can float in here again, and then it can bounce to the next spot, causing the next spot to be activated. So you can see that what happens is, essentially the signal is bouncing in between these areas, between the myelin sheaths. What do we call the area between the myelin sheaths? We call that the node of Ranvier. Remember, these little areas, this would be node of Ranvier, node of Ranvier, node of Ranvier. So basically, with the myelin sheath, we're able to have the action potential kind of bounce, okay? So it's going to be able to bounce from here to here to here without anything happening in this area, okay? So what does it do? What does this do for our nerve impulse? It speeds it up, right? So myelin can conduct neurons with myelin, okay? So some neurons have myelin and some don't. Those that have a lot of myelin, they conduct action potentials much faster because now there's no need to wait for all of these ions to move in here. We can just bounce and move them here, bounce and move them here. So this saves us time and it saves us ATP, energy, right? Because remember, normally these guys, they use ATP to pump sodium and potassium around. Now that we don't have to do that here anymore, we save ATP for these areas, okay? So this whole process of these signals skipping between the nodes of Ranvier, we call that saltatory conduction. So it's the name given to the phenomenon whereby an action potential jumps from one node of Ranvier to the next, okay, along a myelinated neuron. This obviously doesn't happen with neurons that are unmyelinated. So in non-myelinated neurons, the whole the whole axon must undergo depolarization and repolarization. You can't skip these certain areas because there's no myelin. Okay, so that's important. Know what saltatory conduction is and how it speeds up sending of nerve impulses. So now we're done with these three: neuron impulse propagation, IPSPs, EPSP, saltatory conduction. Now we're going to the kind of interesting ones, like more realistic, real life stuff that we need to know about. We're going to learn a little bit about endogenous and exogenous chemicals. What do I mean by that? So, you know, basically in life, you can intake certain things, like you can eat certain medications. Those are called exogenous chemicals because they are made outside of your body and you bring them into your body. So they're exogenous. Exo means outside. And then there are molecules that are already inside your body, right? That your body makes within itself. Okay, so what changes things in terms of the human population is exogenous things. Like we like to create little molecules like drugs, medications, and we like to put them inside our body, right? And they're gonna have certain effects that might be bad or good, right? So we're gonna give you two examples. It's gonna be neonicotinoids and cocaine, and we're gonna see exactly what these exogenous chemicals, how they affect our nervous system. So they're exogenous because they're not made in our body. We have to consume them. So this would be neonicotinoid is one example of an exogenous thing which you can put into your body. Okay. Now in this case, neonicotinoids is an exogenous chemical that we don't put in our body, but we like to give it to insects. So neonicotinoids is a little molecule contained in certain insecticides, and it's the thing that's going to help kill those insects. And why do we want to kill the insects again, right? Because normally you have a nice little plant that you want to grow. So let me put it here. You have a nice little plant, but all these insects on it is killing it, eating it, destroying it. So if you give them these insecticides, hopefully you can kill those insects. And by killing those insects, the plant can grow again. Because it's not the fact, the plant isn't dying because the environment, like the soil, is bad. It's because the insects are eating it, destroying it, okay? So that's why these things are useful. Now, how exactly does this neonicotinoids work? How does it kill an insect? Now, basically, what are we looking at here? We're looking again at a presynaptic neuron and a postsynaptic neuron. So two neurons adjacent to each other. And we know a signal is sent by these little, neurotransmitters, right? They're going to be released here by the vesicles and they're floating across and they want to bind to these receptors. This acetylcholine, right? When it binds, so ACH is an abbreviation for acetylcholine. When it binds to the receptor, that causes this channel, which is ligand gated, because ligand is any kind of molecule. So since acetylcholine is a molecule and it binds there, it's gonna open this channel, okay? When this channel opens, now guess what can happen? Sodium can come rushing in, and that can cause the this neuron to undergo become positive and undergo an action potential, and the positive charge can be spread and the signal can be sent all across the neuron to the next part, right? Now, these neonicotinoids, what they do is they bind to this area where acetylcholine wants to bind, right? So it's kind of blocking it's like basically like taking the seat of acetylcholine. Now that acetylcholine can't bind, guess what? Now sodium can't go in. And if sodium can't go in, then guess what? We can't make a signal, we can't make an action potential, and therefore we can't send the signal further along. Now what does this mean? This means, think about it, this means we can't send signals to our muscles to contract. We can't send signals to our diaphragm, which is responsible for helping us breathe. We can't send signals to our heart, which is responsible for helping us contract and send blood around our body. We're going to become paralyzed. We're going to die, right? So you can see that is how this little guy kills little insects, right? So rest in peace. So basically it's a rest in peace drug, okay? It's pretty cool. It's useful because it helps farmers make better food and be able to survive, right? Obviously, if you take some of this, it may give you some symptoms, but to be able to kill a human, you'd have to take quite a lot of it, right? We're way bigger than these insects. So you will get some symptoms perhaps, but you won't necessarily die unless you take a ridiculous amount, okay? So here's a little summary of that. What's also interesting to note is when these bind, you know, normally, right, when acetylcholine binds, eventually there's like little enzymes that will break acetylcholine down and send it back into the presynaptic neuron to be recycled so that and the next signal can be received and the next signal can be sent, basically recycling the process. The thing about these neonicotinoids is that they're not broken down. They stay there permanently. So if they stay there long enough, you're definitely going to die, okay? So that's it for this one. Let's go on to cocaine, guys. So let's talk about how cocaine, because remember, cocaine is also an exogenous molecule. It's not made in our body. We can consume it from the outside world because it's made by certain people, right? And you can buy it and consume it. It's an exogenous molecule. Now, how does cocaine work? Okay, it's a very, very interesting question because obviously a lot of people consume this and a lot of people get addicted to it, and it's a very, very problematic thing. But many people don't actually know how it works. So and again, it affects neurons, okay? It affects our brain, our nervous system, our nerve cells. So normally what happens is there's a certain neurotransmitter, okay, called dopamine. And dopamine, when it's released, it gets sent across here and it diffuses and it goes to the postsynaptic cell and it binds it. And when it binds it, it allows the signal to be sent. Now, basically dopamine, okay, the signal that dopamine allows to be sent will go to a part of your brain that makes you feel very happy, very fulfilled, very excited, okay, very pleased, okay? It's a very, very positive neurotransmitter. So it causes a signal to be sent to your brain that makes you feel all of these good sensations. Now, that's normal. So when you, for example, do sports, maybe there's a release of dopamine. You get happier, excited. Maybe if you watch a great movie, there's some dopamine. Maybe if you get your first kiss, right? All of these funny little things can release dopamine in your brain and cause you to feel these good sensations. Okay, that's normal. Okay, it keeps us motivated, keeps us happy. But cocaine is doing it unnaturally, okay? So cocaine, if you take cocaine, what it does is it causes these little guys to be released even more, okay? So it's stimulating these vesicles to go and fuse here and release even more dopamine. So if you have even more dopamine, you're going to get even more of these happy feelings, sensations, motivation, and things like that, right? And that's one way in which it acts. Another way is it prevents the breakdown of dopamine. You know, normally neurotransmitters have to be broken down by little enzymes to be recycled so that it can be reused, right? So the next signal can be sent the next time you kiss a girl, right? But this cocaine prevents its breakdown. So it's permanently here, sending big signals all the time. See, I put a big arrow here, okay? So it's acting in two ways, okay? So cocaine increases your dopamine release and prevents its breakdown so that there's a constant sending of this signal to create this sensation in your brain. Now, so that's the mechanism, okay? So you can see here, reward, pleasure, blah, 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 okay? Now, what's the problem with it? Because you might be like, isn't that great? Let's just all do all this. Let's all do it. Let's all do cocaine. Why not? It's making us feel good. It's a huge problem, guys. The problem is, if you take this, okay, and your brain gets used to being this super high and this super excited and happy, the next, if you suddenly stop taking cocaine, now even normal things that used to make you happy will not make you happy anymore. Okay, when you play sports, you will not be as happy. Okay, when you do other things, you won't be as happy, because this thing creates such a positive sensation that it's uncomparable to the things that made you happy before. Okay, and that means people start taking it, and then they start, and it's just like smoking. If you take it for long enough, you start getting used to it. So even taking a gram of it makes you just feel normal. So after a while, these people start taking cocaine just to feel normal again. Okay, and then they need to take even more to feel good. So that's why over time they increase the doses, because they need more to feel happy again. And after a while, they just take it to feel okay, all right, just to feel not depressed. So that's the problem with this thing. Okay, the brain, after time, will start adapting to this unnatural reward pathway and become less sensitive to natural reinforcers, like sports, like just talking to your friend, laughing, making jokes. So this will increase the likelihood of seeking another drug. So other than cocaine, stronger drugs, you'll start going for things that are even stronger. Okay, instead of natural things, right, like relationships, food, and other things like that. And then, like I said, over time, you'll have a tolerance. You'll start becoming used to it. And you need to have more doses and more other things. And it has a lot of other side effects that you don't want, right? Very, very, so that's the thing. So it may look like a good thing on the surface, but just like smoking and alcohol, it has its effects, right? You end up needing it just to feel okay. Okay, so my advice is don't do it. Okay, it's not a good thing. Try to stick happy with the basic things we do in life. As soon as you do this, you're going to depend on it. Okay, so hopefully that's useful to show how exogenous molecules like cocaine and neonicotinoids affect our nervous system to affect the way these signals are sent and makes us feel a certain way, right, artificially. Obviously, guys, there are many medications that we can also take that are actually useful to making people that are depressed feel a bit better and things like that. So these are just two silly examples. There are many, many other examples that can affect our nervous system. It's a really crazy cool, especially when it starts coming to medicine, how it can make you feel better and solve certain problems, okay, certain diseases. All right, guys, we're coming now to the last part, role of receptors. So this one's pretty interesting. So remember, guys, how we have our nervous system is comprised of the central nervous system, our CNS, and the peripheral nervous system, our PNS. And our peripheral nervous system had motor neurons, which are basically neurons sending signals from our brain to our muscles, our glands, and our like all our organs and things like that to make them do a certain job. And then we had our sensory neurons, which do the opposite. It goes from places like our skin or our organs and send messages towards our brain to tell our brain something that happened to our body, like pain and things like that. Now, we're going to look here at some sensory neurons, okay? So neurons sending signals from some place to our brain. Okay, now we have a lot of sensory neurons, different kinds that pick up different signals and send different kinds of messages to our brain. So let's start off here. Look at this one. We have some neurons. By the way, we call these neurons that receive a signal from the environment or some place in our body, we call them receptor neurons, okay? Because they have a little receptor is something that picks up a signal, okay? It recepts a signal, receives a signal. So it's a neuron that converts a physical stimulus of some sort. We're going to talk about all of these and basically converts it into an action potential, right? So it converts this signal into an action potential, this positive charge that will now be sent down this neuron and then to the next neuron, then eventually to our brain, okay? So it's a cool neuron that can convert any of these kind of stimuluses into an action potential and start a signal, okay? So let's look at this one. This one is this one neuron. There's some neurons in our body that can pick up light. We call them photoreceptors. Imagine where these are. Where do you think they are? In our eyes. So we have these these neurons in our eyes that can pick up light so we can see things. So that's pretty cool. Photoreceptors, light. So they pick up light. Then we have these other ones. They're called chemoreceptors. For example, these can pick up some things like smell or taste. So where do you think they exist? In our nose or our mouth, right? That's pretty cool. Okay, then we have baroreceptors. These are some they have little receptors that pick up pressure. So you know your blood pressure is very important. If it's too high, you can get a heart attack or a stroke and things like that. Now there are little nervous system, little neurons in your body that can pick up the pressure in your blood, okay, by little receptors, and it can send that message to your brain. Because if the blood pressure is maybe too low, your brain can then decide to send a signal to increase your blood pressure to keep it normal. Okay, so that's another cool kind of thing. So baro means pressure, so it's a pressure receptor. So all these words directly mean say what they mean: chemo, chemical receptor; photo, light; baro, pressure. Okay, and then electromagnetic, there's some that can pick up electrical currents and things like that. Okay, thermoreceptors, this one's pretty cool. Think about this one. When your skin, when you touch something hot, there are little cells that have these receptors, these little neurons that can pick up temperature, can tell your brain if you're touching something warm or something cold. And then there are other ones, nociceptors. This one's really special. Noci means pain. So these are like pain receptors. So they can, I don't know why, I have a bracket there. So for the sake of all cringing people, I'll remove it just because I'm a nice guy. So, you know, nociceptors are pain receptors, so they can pick up pain. Now, you might be like, my guy, what is pain? Like, what exactly is pain? Define pain. Most things in excess leads to pain. Look, if temperature is in excess, you're going to get burned. That's pain. If someone touches you, puts pressure on your skin, if it's initially it's nice, but if they put too much pressure, it's pain. If let's think of another example. If someone scratches you lightly, it's nice, but if it's too much, it's pain. Okay, so anything in excess is pain. So these neurons pick up anything that's excess temperature. It will start sending that signal to your brain as pain. So you'll experience it and be like, "Ooh, that's painful. Stop doing that." Okay, that's important. So in general, all of these the concept's the same as what we've been learning about throughout this whole video. These nerve endings, okay, these receptors, will basically pick up the stimulus and convert it into a positive charge. Certain channels will open, like sodium channels and things like that, allowing them to float in to make the inside positive. And that will cause an action potential to be sent through the neuron and eventually to where your brain, where the CNS that will interpret it and figure out what that is, and now tries to determine what to do with that information. Okay, so that's interesting. Now, another kind of cool thing with nociceptors that your book mentions is chili peppers or jalapenos, you know, spicy stuff. They contain a little molecule called capsaicin. So it's a little chemical. But what's interesting is this chemical binds to these little receptors here that pick up temperature. So when they bind to it, it will create the signal in these neurons that normally pick up temperature. So now your brain's going to think you're warm, even though it was just a little chemical. So isn't that interesting? So these chilies, they make you feel like you're warm, even though you're not, even though it's just a little chemical, because they bind to the receptors that pick up temperature sensation. So that's pretty cool. That way, it kind of tricks your system to thinking you're warm, right? And obviously, if you eat way too much of them, it's going to activate your nociceptors and make you feel pain, right? So I hope that makes sense. Now, obviously, no, medications is super important, okay? And they can kind of modify the way your neurons are, and I kind of talked about on the previous slide, so I'm not going to talk about that anymore. And what's interesting to know is anything that can go right in your body can go wrong, okay? So there's many congenital issues, which are basically issues that you're born with. So some people, for example, don't have the right receptors, or their receptors or some of their nervous system cells, their neurons don't work as nicely. So for example, some people are born without the ability to feel pain. These little neurons don't have the right receptors, or their receptors don't work well, so they can't pick up pain. So these people will end up breaking every freaking bone they have without even noticing it. And the thing is, that's bad, because they will, for example, hold a very hot pan and not realize it, and then next moment they look at their hand and it's all screwed up. Okay, so you may think it's cool not to be able to feel pain, but trust me, you don't want that at all. You need pain. It allows you to know what's going on, to not break your body completely, okay? So you can stop doing the wrong thing soon enough before you completely destroy yourself. Okay, so guys, that's really it for this video. It was a lot, I know, but now let's go do a couple of questions and answers, and then we're done with this one. So question 1 here, guys, and again, get many more at teachme.org. Which of the following is true regarding saltatory conduction? It reduces conduction rate in a neuron. Is that true? No, right? It increases the conduction rate. We're skipping all of those little areas by having that myelin. It reduces ATP use. That's true. All right. Occurs in myelinated neurons. Yeah. So we know 2 and 3 are correct. So it's going to be, answer is going to be C. So 2 and 3 only. Next. Neurotransmitters are involved during synaptic transmission where they can either stimulate or inhibit a signal transmission. Which row correctly describes the associated neurotransmitters? So GABA and acetylcholine. So remember acetylcholine, right? That was the excitatory one. When it binds, it causes the postsynaptic neuron to become excited and depolarized and an action potential to happen. Whereas GABA, that was an example of the opposite. It was inhibitory. So we're looking here for inhibitory and excitatory. So it's going to be C. One more, guys. What effect does cocaine have on the neurotransmitter dopamine? Remember, cocaine, it did 2 things. Go back to it for you guys. It increased dopamine release into the synaptic cleft, and it prevented its breakdown, okay? So blocks the release of dopamine. No, it actually increases the release of dopamine. So that was not correct. Prevents the removal of dopamine from the synapse. Oh, that's true. Remember, it prevents the breakdown of dopamine. So there's more of it in the synapse. So that one's going to be true. Let's see the rest. Increases the degradation of dopamine. No, it actually does not break down dopamine. It makes it stay there in the synapse. So it's not that one. Binds to the dopamine receptors. No, it does not actually bind to the receptors. Okay, it only influences the amount of dopamine. So it's gonna be B, right? So that's it for this video, guys. I really hope it was useful. We cover a lot, and some of it is really tricky. So I hope it was useful, and I'll see you guys in the next one.